professor of medical laboratory science at Baptist Health Science University. I'd like to show you one of the most important tests that we use in a medical laboratory, and that's the Gram stain. The Gram stain will stain bacteria, and it is important because we can determine two major groups of organisms, whether they're gram positive or gram negative. That's important because we can base that gram stain result on how we're going to proceed with further identification. Once we do know if it's gram positive or gram negative, we can then look at the morphology under a microscope and take that a little bit further. But the most important thing is if it's gram positive or gram negative. How does the gram stain do that? How does it work? We have 4 basic steps to the gram stain. The gram stain is going to stain the cell wall of the organism. Gram positives are different from gram negatives in that the gram positive organisms will retain the primary stain, which is called crystal violet. We'll stain that for about a minute and rinse it. All organisms will be stained with that primary stain. However, after we add in a mordant, which is grams iodine, let that sit for about a minute and then rinse it. We have a third step. The third step is to decolorize. The gram positive organisms will retain that crystal violet primary stain. It has to have the mordant to cement it in place. Basically, you can look at it that way. And then the decolorizer, which is an acetone alcohol solution. When we decolorize, after that step, the gram negative organisms have lost that crystal violet. The gram positives have kept that crystal violet within their cell wall structure. Then we have a counter stain. The counter stain is safranin. It's a red color. Once we stain with the counter stain, the gram negative organisms that have lost their crystal violet color will stain red or pink. It's usually a pink color. And then we rinse. The gram positive organisms will be a dark purple color, and the gram negative organisms will be red. Now let me show you the procedure. For a gram stain, you're going to need a dedicated sink because stains will ruin a sink. It will basically permanently stain your sink in your laboratory. So you need a designated sink, a water source, a tray to stabilize the slides, and a way to rinse. I personally like to use a beaker or some container. Some laboratories will add a tube to the faucet, and that's just so you don't put too much pressure when you're rinsing the stain from the slides. And you don't want to rinse your organisms off of the slide. So this is my preferred procedure. You also want a way for it to drain into the sink. You want your stains, your crystal violet, your grams iodine, your decolorizer, which is acetone alcohol, and your safranin. You also would need a timer to time 1 minute each step. You need some bibulous paper, and this is helpful when you are done with your staining. You can just put your slide in the bibulous paper and blot it dry. You never want to wipe your slide clean. And some people like to use tweezers or even a clothespin. So let's start. We have our slide. We have put our organism that we're wanting to gram stain on the slide. We've also let it dry, and we've heat fixed it. The heat fix step seals those bacteria onto the glass surface, and they won't wash away at that point. They're all dead now. There's no risk of being contaminated with your hands or infected, but I use gloves anyways. So I'm going to place my slide on my rock. I'm going to add, we call it flooding the slide. You don't need much stain. You just flood the slide. And you set your timer for 1 minute. After that, I'm going to rinse that stain, and then I'm going to add my mordant. Let that sit for 1 minute, and that's going to cement that stain, crystal violet stain, into the organisms. They'll all be gram-positive at that point, but then I'm going to add my decolorizer. That's the most important step of the gram stain procedure. If you decolorize too much, your crystal violet, even though it's gram-positive and should retain that stain, it will actually be overcome and wash away. So then you'll get an incorrect result when you're reading your slide under a microscope. We look at bacteria under an oil immersion scope field, and that's 1,000 times. Our objective is 100, and our ocular is 10. So we multiply that, and we have 1,000 times. If you don't decolorize enough, your gram negatives could look gram positive, so we don't want that either. So the decolorization step is the most important step, and it does take practice. I have already finished with the first part of the stain, the primary stain. Let it sit a minute and rinsed. I've added my mordant. I've let it sit for 1 minute, and I'm about to rinse. When this timer goes off, it's time to rinse. This is my slide here, and this is the iodine, the mordant. So it's complete. I'm just gonna rinse, and that's all it takes. I like to take a little bit of the water off of my slide, and I'm going to decolorize now. So this is the step. You see the purple coming off? You don't want to decolorize too much. It's only about 5 to 10 seconds. That's all it takes. And then you rinse. Now it's time for the counter stain, the safranin. What color are gram-negative organisms going to be? What color are gram-positive organisms going to be? So we're going to let that sit for about a minute. But for the sake of time, we're going to pretend that it's been a minute. It's time to rinse again. So it's a very easy procedure, and it's one of the most important procedures we use in a medical diagnostic lab. Now the fun begins. We've completed our gram stain. We've blotted it dry. We did not swipe it or wipe it. We would lose our little organisms. We're going to look at it under the microscope now. So the most important tool we have in a medical laboratory is the gram stain. We always go back to the gram stain. If we have problems, we will go back and look at the gram stain. It's a great troubleshooting tool. Once we know the gram stain result, gram positive or gram negative, we can take it even further and differentiate our bacteria even further and look for those pathogens and help the physicians to treat our patients. The most important takeaway is how the gram stain works. The gram stain works because gram-positive organisms have a thicker layer of something called peptidoglycan in their cell walls. And the gram-negatives have a thinner layer of peptidoglycan. So the principle of the procedure, the gram-positives will retain more of that primary crystal violet even after the mordant is added and even after decolorizing. The gram-negatives, since they don't have as much peptidoglycan in those cell walls surrounding the little organisms, they will lose that crystal violet even after the mordant and the decolorizer procedure. And then the counterstain is needed because we need to be able to see the gram-negatives. Otherwise, we wouldn't be able to see them. Gram-positives are going to be purple and gram-negatives are going to be red. Thank you for watching this video. I hope you enjoyed watching it as much as I have enjoyed making it.